Hello children, welcome to Vivid Botany. We have a question here about the formation of glycoproteins and glycolipids. Glycoproteins, glycis means glucose. So glucose is nothing but carbohydrate. So when proteins are associated with the glucose carbohydrate, it is called as glycoprotein. When lipids are associated with the glycis carbohydrates, it is called as glycolipids. And glycoproteins, they have so many functions that they enable the sperm to attach on surface of the egg cell. Glycolipids, they provide energy to cell, a source of energy. You know that gives rise to ATP upon the respiration. And glycolipids are the constituents of cell membrane helpful in knowing blood group. We know AB blood groups, O and all that, they are explained by, detected by the glycolipids. And coming to endoplasmic reticulum, it is the network of tubular structure proposed by K.R. Porter. It is a single membrane system, part of single membrane system, single membrane enclosed by part of endomembrane system. This ER with a ribosome, it is called rough endoplasmic reticulum. Without ribosome, it is called as smooth endoplasmic reticulum. Rough endoplasmic reticulum is helpful for protein synthesis. Smooth endoplasmic reticulum is helpful for lipid synthesis. Apart from that, rough endoplasmic reticulum, I usually call it as rare. The rare is helpful for secretion and SER is helpful for lipid-like steroidal hormones synthesized in animal cells. This is. And coming to peroxisomes, they are enclosed by only one unit membrane. They are microbodies. They are found in both plants and animals, discovered by Rodin. They cause photorespiration, is also called as glycolytic pathway, is also called as the C2 cycle that takes place only in C3 plants but not in C4 plants. During photorespiration, that rubisco acts as oxygenase and rubisco accepts the oxygen to give rise to the glycolic acid as well as glycerin, phosphoglyceric acid. So in this process, CO2 liberates and the oxygen is absorbed. So it is respiration takes place in the presence of sunlight and the loss of carbon dioxide by consuming ATP. So it is considered as a waste process in plants. Fortunately, it is not found in C4 plants, it is detected only in C3 plants. Apart from that, peroxisomes also cause breakdown of hydrogen peroxide into oxygen and water. You know, hydrogen peroxide is toxic to the living plants. So, this hydrogen peroxide is broken down by peroxisomes and it will help the plant to protect from the toxic effects. And synthesis of phospholipids is also done by the peroxisomes. Then coming to polysomes. Polysomes are ribosomes present in a row on mRNA. They translate genetic message present on mRNA and they cause the synthesis of amino acids by peptide bonds to form the polypeptides that are proteins. So the main function of ribosomes is to synthesize proteins. Coming to Golgi bodies. Golgi bodies are densely stained reticular structures found nearby nucleus. They are reported by Camillo Golgi, hence they are named after the scientist Golgi bodies. They are packaging intracellular material. Their main function is glycoproteins and glycolipid synthesis. Apart from intracellular packaging, after intracellular packaging, they may release that packed item either inside the cell or outside the cell. So if you see the question here, which is the important site for formation of glycoproteins and glycolipids in eukaryotic cell? Endoplasmic reticulum. Endoplasmic reticulum will synthesize lipids and proteins, but not glycoproteins and glycolipids. That is the most important point. So it is not the answer. Then peroxisomes. Peroxisome synthesis phospholipids, not the glycolipids. I wrote here. They synthesize phospholipids but not the glycolipids. And peroxisomes are responsible for the breakdown of hydrogen peroxide, for occurrence of the C2 cycle or photorespiration. So this is also not the answer. Then coming to polysomes, that are ribosomes arranged in a chain on mRNA are called polysomes or polyribosomes. They are meant for translation of genetic message present on mRNA to form the proteins. So they don't make up glycoproteins and glycolipids. So these are the NEET answer. And coming to Golgi bodies or Golgi complex. You know, Golgi bodies or Golgi complex is also called a lipochondria. Is also called as lipochondria. And another term, that Golgi complex also called as idiosome. Idiosome. They are also called as Baker's bodies. Baker's bodies. So these many names are there for Golgi bodies: Baker's bodies, idiosomes, lipochondria, Golgi complex, Golgi bodies. And Golgi complex or Golgi bodies having two phases, cis and trans. Cis is convex phase, trans is concave phase. So it is a bipolar cell organelle. It is something different than the rest of the cell organelles, but though the two surfaces are different, but they interconnected with each other. And these Golgi bodies are meant for intracellular packaging apart from synthesis of glycoproteins and glycolipids. So the answer is here, the Golgi bodies that will cause synthesis of glycoproteins and glycolipids. So please subscribe my channel Vivid Botany, and you have any doubts, make a comment, drop a comment in the comment box on my channel Vivid Botany, so that we can discuss, you can clarify your doubts in our next video. So thank you very much.